Every biologically manufactured product is at risk of viral contamination, which can have serious clinical and economic implications. Sources of viral contamination are among other raw materials, equipment, cell banks, or breaches in cGMP production. Major concerns are given by viruses endogenous to the cell line and adventitious viruses. To guarantee patient safety, regulatory authorities have defined three principal approaches for viral safety. 1. The selection of clean source material. 2. The implementation of virus clearance steps. 3. The testing for known viral contaminants. Virus clearance steps must be part of all biologics manufacturing. Virus filtration, low pH inactivation, chromatographic steps. To prove the effectiveness of each purification step, virus clearances studies must be conducted. Before any virus clearance study, a model panel of viruses with a variety of physiochemical characteristics must be chosen. Every study must represent a valid scaled-down model to guarantee reliable comparison between the manufacturing and the laboratory processes. Virus clearance studies can be divided into pre-study, main study, and reporting. Viruses are spiked into the test material. The test material is filtered through the virus filter at constant pressure. Critical process parameters are monitored. Samples are taken during the process step and assayed for viral infectivity. These samples are serially diluted. Positive and negative controls are included on each day to ensure assay robustness. To determine the effectiveness of the virus clearance step, the log reduction value needs to be calculated. The LRV is calculated by comparing the virus titer before and after the performed purification step. Virus titers are determined by a TCID50 assay, in which the cytopathic effect, CPE, is observed and evaluated at various dilutions of the test samples. CPE indicates cell death caused by the infection of the virus. Once the main study is finished, data is compiled in a virus safety report. To guarantee a reliable result of the TCID50 assay, pre-tests are performed. This consists of cytotoxicity and viral interference testing, which are conducted prior to the main study. Cytotoxicity and viral interference tests ensure the test material does not impact on cellular or viral growth, therefore avoiding unreliable LRV results. In addition to careful study planning, three key aspects must be considered for a successful virus clearance study. First, correct product handling, such as proper wetting and equilibration of filters and membrane adsorbers. Second, purified virus stocks to prevent fouling of filters and membrane adsorbers, which could reduce the clearance effectiveness. Third, valid scale-down model to guarantee reliable comparison between the manufacturing and the laboratory processes. We can study different products under various conditions. Confidence. Virus Clearance Services gives you the confidence you need to succeed.